That's it, Molly. Module 2, Basics of Mendelian and Mitochondrial Inheritance. Mendelian inheritance. Mendelian characteristics are determined by chromosomal loci, either on an autosome, which recall are human chromosomes 1 through 22, or on a sex chromosome, which could be either X or Y, depending on gender. Females are diploid at all loci and have 23 pairs of homologous chromosomes. Males are different. Like females, they have 2 copies of each autosomal locus and of pseudoautosomal sequences, which are found at the tips of the sex chromosomes. However, they are hemizygous for the great majority of the loci on X and Y. Males have only 1 copy of the majority of loci that are located on the X and the Y, but outside the pseudoautosomal regions. As a result, there are 5 basic Mendelian inheritance patterns: autosomal dominant, autosomal recessive, where you could have a situation where someone is a carrier, and you can also have individuals who are compound heterozygotes, and this would indicate that they would have one variant on one homolog and another variant on their other homologous chromosome. We can also have characteristics of X-linked recessive and the X-linked dominant, as well as the Y-linked. Autosomal dominant pedigree. A dominantly inherited disorder is one that is manifested in heterozygotes. Affected persons usually carry 1 mutant allele and 1 normal allele at the disease locus. In autosomal dominant inheritance, the disease locus is present on an autosome, any chromosome other than the X or the Y, and so an affected person can be of either sex, as is shown in this pedigree. Clearly, you can see we have males as well as females who are affected by the disease. We know they're affected because, as you recall, the filled in circles or filled in squares means that these are affected individuals. Both sexes are affected and equally likely to transmit the disorder. When an affected parent has children with an unaffected person, each child would typically have a 50% chance of developing the disease. Affected individuals are typically heterozygotes with 1 mutant allele and 1 normal allele, and usually have at least 1 affected parent. The question mark indicates the chance of having an affected child, which is 1 in 2, because 1 parent is affected. There's a 50% chance of transmitting the mutant allele and a 50% chance of transmitting the normal allele. So that's why when we have over here between 7 and 8, and that joining shows that those individuals have a 50% chance of having a affected child. Autosomal recessive pedigree. A person affected by an autosomal recessive disorder can be of either sex and is typically born to unaffected parents who are heterozygotes. The parents would be described as asymptomatic because they are carriers of the mutant allele, and they have 1 wild type allele, and thus they are unaffected. Affected individuals carry 2 mutant alleles at the disease locus, 1 inherited from each of the parents. Assuming that both parents of an affected child are phenotypically normal carriers, The chance that each future child born to these parents is also affected is normally 25%. The risk that one parent transmits the mutant allele is 1/2, or 1/2. So the risk that both transmit the mutant allele to the child is 1/2 times 1/2 equals 1/4. And that calculation is based on standard probability mathematics. Every one of us carries a single harmful allele at multiple loci associated with recessive phenotypes. When an autosomal recessive disorder is quite frequent, carriers will be common. In that case, an unaffected child may be born to two parents who carry different mutant alleles, and the affected individual with two different mutant alleles is described as a compound heterozygote. This slide depicts an autosomal recessive pedigree. In A, we have a standard autosomal recessive pedigree, and in B, we have an autosomal recessive pedigree in a case which involves consanguineous relationships. In A, the pedigree for a common autosomal recessive disorder is shown. The parents of the affected children in generation 4 are carriers with one normal allele and one mutant allele. If they are not known to be related, they might well have had different mutant alleles. From the pedigree alone, we would not know who carried mutant alleles in generations 1 and 2. For each subsequent child of 3-2 and 3-3, the risk of being affected is 1 in 4, irrespective of the sex. In B, we show a scenario involving consanguinity. Here we know that 3-2 and 3-3 are first cousins. They can be expected to be carriers with one mutant allele M and one normal allele N. We could infer that 2-2 and 2-4 inherited the same mutant allele, shown in red, M, from one parent, either 1-1 or 1-2. That means that 3-2 and 3-3 have the same mutant allele, and that their affected children will have inherited two identical copies of the mutant alleles and be true homozygotes. The chance that their fourth child may be affected, which is shown by question mark, remains 1 in 4, irrespective of its sex. This is a good slide to take a look at and make sure that you understand the relationship. So remember that the older generations are shown at the top, and as we move down the diagram, we move into the most recent generations that are shown in 4. X chromosome inactivation. This slide helps to illustrate the mechanism that compensates for the variable number of sex chromosomes in humans. Females have 2 X chromosomes and males have 1 X and 1 Y. Chromosomal number is highly regulated, and the consequences are typically lethality that occur with the loss of just one of our 46 chromosomes, except in the case of 445X, which is Turner syndrome. Having extra chromosomes is often disastrous as well. It can either be lethal or result in profound developmental defects, such as the case in trisomy 21 or Down syndrome. This happens because of the problems associated with gene dosage. So the number of chromosomes in turn regulates the number of loci that are present, and that in turn regulates gene dosage. The sex difference between the Y chromosome is minimized by the conspicuous lack of genes on the Y chromosome. Most of the very few genes on the Y chromosome have male-specific functions, or they have an equivalent gene copy on the X. These XY gene pairs are largely concentrated at the tips of the sex chromosomes and are referred to as pseudo-autosomal regions. So unlike the Y chromosome, the human X chromosome in females has many important genes. To compensate for having different numbers of X chromosomes in males versus females, a special mechanism is needed to silence the genes on one of the two X chromosomes that each female has. This process is referred to as X inactivation. That is to say, men are constitutionally hemizygous for most genes on the X chromosome, and women are functionally hemizygous for the same genes. This slide depicts X-linked recessive inheritance. Affected males, as shown in A, in generations 3 and 4 have inherited via female carriers a common mutant allele from 1-2. In each case, the child of a carrier mother, such as 3-6, the overall chance of being affected is 1 in 4. But this is sex dependent. A son will have a 1 in 2 risk, but a daughter will not be at risk, though she has 1 in 2 risk of being a carrier in the highlighted box. Three, 3, and 3-4 have 2 normal children, and the risk of having affected children would normally be very low. The father cannot transmit the mutant allele, which is on X, to his sons. He must transmit a Y, and any daughter inherit a normal X from her father. In B is shown the complications of inbreeding on a recessive allele. Imagine that 3-3 and 3-4 were consanguous and they had the same mutant allele. We now have a mating between an affected individual and a carrier, and there is a 1 in 2 chance that a child would be affected, irrespective of whether it was a boy or a girl. The apparent male-to-male transmission is an illusion. The affected son has actually inherited a mutant allele from his mother, not from his father. The affected daughter is homozygous, and although 1 mutant allele will be silenced in each of her cells, she does not have a normal allele. This slide shows a pedigree depicting dominant inheritance as shown in females. This slide depicts each child of an affected parent having a 1 in 2 chance of being affected. Remember, affected individuals are shown by the filled symbols. There's a 50% chance that an affected female can transmit a mutant X allele to sons and to daughters. The risk is the same, irrespective of the sex of the child. However, the risk to the children of an affected father depends crucially on the sex of the child. Every son would be expected to be unaffected. Instead, the risk of being affected is focused on daughters, as shown for the 3 affected males in generation 3 who each had children. The father must pass on a Y chromosome to each son, and so does not transmit the mutant X, but must transmit the mutant X chromosome to each of his daughters. This slide depicts X-linked dominant inheritance in males by pedigree. X-linked dominant inheritance with early male lethality. This example shows 4 affected females in a 3-generational family with incontinentia pigmenti that was followed up after the birth of an affected granddaughter, shown by an arrow. An affected male in generation 2 had spontaneously aborted. This slide also depicts some interesting phenotypic features of X-linked autosomal dominant inheritance. In autosomal dominant disorders, affected individuals with X-linked dominant disorders can be of either sex, and usually at least one parent is affected. However, there are significantly more affected females, as shown in this pedigree, than affected males. This is because there is no male-to-male transmission of the disorder. All children born to an affected mother and an unaffected father have a 50% chance of being affected. But an affected father with a single X chromosome will consistently have unaffected sons, since they don't inherit his X chromosome. So his daughters will always be at risk because they always will inherit his affected X chromosome. The milder phenotype seen in affected females is a result of X chromosome inactivation. The mutant allele is located on, if the mutant allele is located on an X inactivated X, the proportion of their cells that have this disorder will be limited. All affected individuals are female, so the phenotype is so severe that they often die in the prenatal period. But the milder phenotype of affected females allows them to survive and reproduce. This is illustrated in this slide. The human X and Y chromosomes, differences and major homology regions. The X and Y chromosomes differ greatly in size, heterochromatin content, and DNA sequence. In this figure, colors indicate sequences that are X-specific in pink, Y-specific in blue, heterochromatic and hatched, or shared by the X and Y chromosomes only. Other colors. The major pseudoautosomal regions on the X and Y in yellow are essentially identical, as the major pseudoautosomal regions on the long arms are green. The pseudoautosomal regions are involved in the XY pairing and recombinations in male meiosis. The large central regions of the X and Y do not engage in recombination and are X-specific or Y-specific. As a result, an evolutionarily recent XY transposition event, there is also a roughly 99% sequence homology between sequences on Y close to the major pseudoautosomal region on X, shown by purple bands. This slide depicts pseudoautosomal inheritance and XY recombination. In A, in the pedigree, affected individuals are heterozygous for a mutation in the major pseudoautosomal region, as we showed you in the previous slide. And the disorder shows dominant inheritance as a result. Affected females carry the mutation on an X chromosome, which can be passed to both sons and daughters. Affected males pass a Y chromosome containing the mutant allele to affected sons, but can also pass an X chromosome containing the mutant allele to affected daughters. This happens as a result of the obligatory XY recombination, which occurs within the major pseudoautosomal region. In B, when the crossover point occurs proximal to the mutant allele, the allele is transposed between the X and Y chromosomes. Matrilineal inheritance and mitochondrial DNA. The mitochondrial genome is a small 16.5 kilobase circular genome that has 37 genes. This DNA is much more prone to mutation than nuclear DNA, partly because of its proximity to reactive oxygen species that are present in the mitochondrion. Individuals with a mitochondrial DNA disorder can be of either sex, but affected males do not transmit the condition to any of their children. This is because mitochondria are inherited maternally. Only the maternal lineage mitochondria are present in offspring. Recall that several hundred to thousands of mitochondrial DNA are present in a given cell. And each cell contains multiple mitochondria, which gives rise to this fact. But affected individuals have cells with mixed populations of normal and mutant DNA because of the phenomena of heteroplasmy. So remember that when the mitochondria divide, they do not partition their mitochondrial DNA evenly. So if there are variants, these are not going to be dispersed evenly to the offspring mitochondria. This phenomenon is known as heteroplasmy and was discussed in earlier chapters. This slide illustrates matrilinear inheritance from mitochondrial DNA disorders. Mitochondrial DNA disorders are transmitted by females only because mitochondrial DNA originating from the sperm is quickly degraded in the early embryo. An affected female can pass on the condition to both sons and daughters. A common feature of mitochondrial disorders is incomplete penetrance. That means not a full manifestation of the phenotype according to a given genotype, as is shown here by the absence of the clinical phenotypes in several individuals who must be gene carriers, including 3 clear carrier females in generation 4, each of whom were born to an affected mother and went on to produce affected children of their own. One cause of this intrafamilial variability is variable heteroplasmy. The mutation here was shown to be a nucleotide substitution in the mitochondrial 1,2S ribosomal RNA, or the 12S ribosomal RNA, that was associated with variable hearing loss.